For decades, the senile plaque has been the universally recognized hallmark of Alzheimer's disease, a dense cluster of protein visible in the brain tissue of every patient. The standard operating assumption treats these plaques as extracellular precipitates. In this model, the cell secretes a toxic peptide that gradually clumps together in the space between neurons, much like minerals forming deposits in a cave. But in 1990, a discovery threw this model into question. Researchers found that these plaques were packed with highly active lysosomal proteases. A lysosome is the cell's internal waste disposal unit. It is a membrane-bound sac filled with digestive enzymes designed to break down proteins strictly inside the living cell. If plaques form in the empty spaces outside the cell, there is no clear reason for them to contain the cell's internal digestive machinery. This anomaly suggests the plaque is the structural residue of a cellular explosion, the debris left behind when a neuron dies from the inside out. This observation forms the basis of Ralph Nixon's 36-year research program, known as the endosomal lysosomal autophagy account, or ELA. The ELA thesis argues that Alzheimer's begins as a failure of the cell's internal waste disposal network. We can contrast this with the traditional amyloid cascade model. On the left, a linear sequence. The protein is cleaved, the peptide aggregates, and the plaque kills the neuron. On the right, the ELA model, a cyclical failure where a disposal jam ruptures the cell, leaving the plaque behind. Pharmaceuticals have spent billions targeting the steps on the left, attempting to clear the peptide or the plaques themselves. While the field now broadly accepts that cellular clearance fails in Alzheimer's, the data suggests we have misdiagnosed exactly where in the disposal pipeline the blockage occurs. By misreading a downstream roadblock as an upstream deficiency, we have designed drugs that may do the opposite of what the neuron requires. Treating the entry point of the pipeline while the exhaust is blocked forces the neuron toward an earlier collapse. To see why, we have to look inside the intracellular landscape and track how the cell processes its cargo. The first chamber in the disposal pipeline is the early sorting endosome, where incoming materials are organized for transport. This specific compartment swells to abnormal sizes in Alzheimer's patients, decades before the first plaques appear. The driver of this swelling is a molecular fragment of the amyloid precursor protein called β-CTF. This molecule is a residual fragment left embedded in the membrane, a species separate from the amyloid beta peptide found in the plaques themselves. When β-CTF levels rise, the fragment recruits an adapter protein that locks a critical control switch, the Rab5 protein, into a permanent on position. As a result, these endosomal compartments fuse together uncontrollably, creating a logistical grip lock at the very start of the clearance system. The pathology begins not with a toxic peptide clumping outside the cell, but with a fragment that physically breaks the cell's internal sorting mechanics. Moving further down the cellular pipeline, we reach the terminal chamber, the lysosome. The lysosome can only dissolve waste if its internal environment is highly acidic, typically at a pH of 4.0 to 4.5. This acidity is maintained by a rotary pump called the v-ATPase, which pulls protons in to keep the digestive environment active. In the Alzheimer neuron, the beta-CTF fragment binds to this pump, wedging into the mechanism and halting its rotation. Without the pump's constant activity, the lysosome's pH begins to rise. Once the environment becomes too alkaline, the digestive enzymes inside become inactive. They can no longer break down the proteins they receive. Cellular waste continues to enter the lysosome, but stays there, undigested, creating a massive backlog that the cell cannot clear. The cell is now trapped, starved of the recycled nutrients it needs to function, and choking on the waste it cannot incinerate. This gridlock does not affect all brain cells equally. It requires a specific set of biological conditions to become lethal. The deadly conjunction occurs when a cell has a failed proton pump and is also manufacturing a high volume of amyloid cargo. This profile matches the glutamatergic pyramidal neurons of the cortex, the very cells lost in the greatest numbers to the disease. As undigested cargo accumulates, the cell undergoes a grotesque transformation called panthos. Swollen, deacidified compartments pack the cell body and bulge against the membrane, creating a corona that resembles the petals of a poisonous flower. Eventually, the volume of undigested waste exceeds the strength of the cell membrane, causing it to violently rupture. As the neuron disintegrates, it spills its internal digestive enzymes and its undigested amyloid core into the extracellular space. This inside-out rupture accounts for the 1990 paradox. The senile plaque contains lysosomal enzymes because it is the spilled contents of an exploded cell. If we look at the macro behavior of a neuron trapped in this cycle, we see a specific structural signature. We can divide the cell's disposal process into two legs, a forward leg for input and induction, and a return leg for output and clearance. In the Alzheimer neuron, the forward leg is accelerating. The cell turns up autophagy induction and clears surface receptors faster. But the return leg is paralyzed. Acidification fails, so the central waste reservoir fills faster than it empties. If this were a case of generalized cellular aging, both legs would likely slow down in tandem. Instead, the cell realizes it is clogged and desperately turns up its induction to try and clear the mess, only to add more cargo to a system that cannot vent. The neuron dies because its frantic attempt to increase input collides with a paralyzed exhaust system. This systemic asymmetry reveals a misalignment in current pharmacological strategies. The current industry standard focuses on drugs that induce autophagy, compounds designed to sweep the cell clean by turning up the induction end of the pipeline. But we must weigh this strategy against the state of the Alzheimer neuron. Autophagy induction in these vulnerable cells is already pathologically high. If we give an autophagy inducer to a patient whose lysosomal pumps have already failed, we create a physical catastrophe. The drug does not clear the waste. It merely forces the cell to fill itself even faster with transport vesicles it cannot possibly digest. By ignoring the imbalance between the forward and return legs, current clinical approaches risk accelerating the very rupture they are intended to prevent. The science dictates a specific, biologically sound pharmacological sequence. First, we must restore degradative capacity. This means re-acidifying the lysosome and supporting the v-ATPase pump. Only after the exhaust system is functional can we safely administer drugs that increase autophagy induction. To date, the only drug derived from this framework, neflamapimod, failed in Alzheimer's trials, largely because it targeted an upstream signaling lesion rather than the downstream acidification failure. However, even a perfectly sequenced trial faces a significant technological barrier. We currently have no PET ligand or imaging modality capable of measuring the pH of a living human neuron's lysosome. Without that measurement, we cannot select the right patients for re-acidification therapy or prove that our drugs are engaging the target. Until we can measure the acidity inside a living brain cell, clinical trials are essentially flying blind, treating the wreckage of the disease while missing the engine that drives it.